Hello biologists, this is Miss R and we're going to talk about how to do Lab 3.19. We're going to start where we left off in Lab 3.18 on page 3 of the simulation, thinking about why enzymes are important and what exactly does catalyze mean. The big question here on page 3 is what role do enzymes play in glycolysis? And we can see, because we counted them before, there are 10 of them. They're right here as you go between reactions. And when you catalyze a reaction, you make it happen faster without using any energy. Let's click on one of these reaction arrows and look at some of these enzymes. They're pretty cool. We can manipulate our view of the enzyme by saying highlight changes, and we can do cartoon. And if we zoom in, so we are looking at the active site where it holds glucose here, this enzyme hexokinase is going to hold glucose so it can react with the ATP to pick up an extra phosphate here. So the enzyme is very important in holding the molecules in a particular conformation, in a particular way, in a relationship to each other, so that they can react. And that's how they catalyze the reaction. They make the reaction happen faster. We talked about that in class when we said it's easier for you to meet a friend at the state fair or a mall or a big public place if you both say where you're going to meet if you have a spot. If you just wander around in the crowds looking for each other, you're probably not going to meet up. But if you have a place to meet, like a coffee shop or a particular bench, you're much more likely to find each other. And that's what enzymes do. They connect or sometimes disconnect chemicals and make that happen faster by providing a place for the reaction to happen. Let's look at another enzyme here. We'll look at the active site, turn on cartoon, and we can see that here's this is a product here. This is the end of the reaction. Products are ends of the reactions. Reactants are the beginnings, the ingredients. You can see this is a product here in the active site. G6P has turned into F6P here. We can highlight some changes, so you can see what changed. And this is the enzyme, again, having a special spot that's holding the reactants so they can turn into products. So for number 1, we've talked a little bit about what catalyze means. For number 2, a screenshot of the enzyme hexokinase is shown below. We looked at that. What role is glucose playing in this reaction? I'll give you a hint. It's right here. What happened to glucose as a result of the enzyme? So here's glucose as a reactant, and here's glucose as a product. It's now G6P because it's gained a phosphate. Let's look at number 3. A screenshot of the enzyme fructose biphosphate atolase, this ACE ending tells us that this is indeed an enzyme, is shown below with GADP in the active site. What role is GADP playing in this reaction? So is it on the reactant side or the product side? I'll let you figure that out. And what happened to F16BP, or this molecule over here, as a result of the enzyme? Well, it looks like this kind of got chopped in half. You can see that there's 1, 2, 3, 4, 5, 6 carbon molecules kind of all strung together in a chain. And now we have 1, 2, 3, and 1, 2, 3. So glycolysis happens in the cytoplasm of the cell, and it's our mission now to figure out where the Krebs cycle happens. We'll go to the simulation in a minute. We're going to look at all the reactions in the Krebs cycle. We're going to keep a tally of how many high energy molecules are produced during the process. We'll look at the process as a whole, but I wanted you to have kind of a recap of all the things that we're looking at so it would be easier to keep track of these high energy molecules like NADH, ATP, FADH, and then you can figure out how much energy is transferred into those high energy molecules as a result of the Krebs cycle, the last half of cellular respiration. So the big question on page 4 here is how many ATPs can be made by further breakdown of pyruvate in the Krebs cycle. Remember, in glycolysis, kind of the end of glycolysis is this molecule called pyruvate. And pyruvate is happening, it's the end product of glycolysis. It's happening in the cytoplasm. That's always sort of the gooey stuff that's in between everything in the cell. When we go to the Krebs cycle, the last half of cellular respiration, the last part of making ATP, transferring the energy of glucose to other molecules called ATP, that happens in the mitochondria. They look like little beans. And we'll get some more details on what those little beans do here in a moment. If we scroll down, we can see the Krebs cycle. There's lots of different reactions. We'll look at the first one here. This first reaction where we're breaking down pyruvate, which is coming from glycolysis, it produces one NADH molecule. And that's a high energy molecule we're going to use later to produce some ATP. When we look here at the next step, we don't produce any high energy molecules. But if we go to the next step, no more high energy molecules. We'll look here. Oh, we get another NADH. So far we're up to 2 NADH if you're keeping track on your tallies. There's another one. There's a GDP. There's an FADH2. Remember, there's a spot for that on your tally sheet. And there's another NADH, and then we start the cycle over again. Got a little lost as we went around this reaction cycle. That's okay. All the reactions are on your lab sheet, kind of recapped for you, so it's really easy to look at how many high energy molecules were created. And I've also given you a big hint right here. We can check the answer here for NADH. Remember, one's created as pyruvate, kind of comes into the Krebs cycle here, as well as 3 in the Krebs cycle. How many GTPs? Hopefully you remember. And how many high energy FADH2 molecules are produced for each pyruvate in the reaction above. So hopefully you were able to use these screenshots and the tally as we run around the Krebs cycle to keep track of how many different high energy molecules were produced. Remember, you want to look for the yellow flashy symbol for a high energy molecule. So we looked at the bottom of the sim for how many high energy NADH molecules are produced. We also looked at how many high energy GTPs and how many FADH2s are produced. If you got lost, you can look up here. Now we have to do a little bit of accounting. Assume each NADH will produce 3 ATP, each DTP will produce 1 ATP, and each FADH2 will produce 2 ATP. How many total ATP can be produced by the high energy molecules created in the Krebs cycle? So let's remember how many NADHs were created. That's 4. So we need to multiply 4 times 3. That's for NADH. And we're going to add that to what we get for GDP. I'll highlight all of this in green here so it's easier to see. GDP, we know we'll produce 1 ATP, and we only made 1 GDP, so that's just 1 times 1. That's easy math. And then we have to figure out how many FADH were produced. That was 1. So we're going to go 1 times 2, as each FADH2 will produce 2 ATP. And we only made 1 FADH. So if we go 4 times 3, that's 12, plus 1, plus 2, we get 15. Oops. So there's our answer for number 8. This question is also on the sim, so we can check our answer. How many total ATP can be produced by the high energy molecules? I put in our answer 15. We're going to check our answer, and look, we're correct. And here it helps us with our math a little bit more clearly. 4 NADH times 3 equals 12. 1 GDP times 1 equals 1, and 1 FADH times 2 equals 2. So for the next question, it says that we've seen glycolysis produce 6 ATPs in the making of 2 pyruvate molecules from glucose. And remember, both of those pyruvate molecules go into the Krebs cycle. You've also calculated how many ATPs are produced for each pyruvate that enters the Krebs cycle. How many total ATPs will be created by breaking down 1 glucose molecule? It's very tempting to say, okay, 6 from glycolysis plus 15, that's 21. Let's see if that works. No, because we've got 2 pyruvates from glycolysis coming into the Krebs cycle, so we do 2 Krebs cycles for every glucose. So we actually get 15 times 2 plus 6. So 15 times 2 plus 6 equals 36. And here's a better accounting of that. So in glycolysis, we make 6 ATP and 2 pyruvates. Those 2 pyruvates come in and make 2 NADH. Well, that actually happens in glycolysis here. And then the Krebs cycle produces 2 pyruvate molecules. We get 6 NADH, 2 GDP, and 2 FADH. Remember, they're counting the Krebs cycle twice because 2 pyruvates are made from glucose, and so you do the Krebs cycle twice for every glucose. So we get a total of 36 ATP. That's a lot of energy available from just 1 glucose molecule. So we've just answered question 9 here. How many total ATPs will be created? Now we have to think about which high energy molecules become part of the electron transport chain in the mitochondria. We actually haven't made any ATPs yet in the Krebs cycle. We make the ATPs when we go to the electron transport chain, which is part of the inside of the mitochondria. And I'll show you a little bit more about what that means. So below, here's a big hint for which energy molecules become part of the electron transport chain. And if you're wondering what the electron transport chain is, we'll see it right now. If you're following along in the sim, I skipped over page 5 quickly, and now we're on page 6. I gave you a screenshot from page 5 that should help you out. Page 6 shows us the electron transport chain. This is inside the mitochondria, in the membrane here, inside the mitochondria. that's actually going to use electrons, trade some electrons, and we'll see that in a working animation here in just a moment. And then it's going to push protons across the membrane, and those protons are going to make ATP synthase work. So let's go watch the electron transport chain in action. So in the electron transport chain, we get to watch some of these high energy molecules that we made in the Krebs cycle get put to use trading electrons here. Electrons are represented here by the blue stars. And the more kind of points on the star, the more energy the electrons have. Remember, we looked at the energy levels of electrons when we did photosynthesis in lab 3.18, part 1 of 3.18. And here we have some higher energy electrons that are up on the energy levels, and we have some lower energy electrons. Let's look at each of these protein complexes. It oxidizes NADH to NAD plus and H plus, transferring the NADH electrons to ubiquitone, that's the UQ. Notice that the electrons lose some energy when they go from NADH to UQ. This decrease in electron energy is matched by an increase in the electrical energy that occurs when a hydrogen ion is pushed across the membrane. So you can see these hydrogen ions are being pushed across the membrane. You'll see little yellow channels appear, and then watch this here. You get energy from the H plus concentration difference, these hydrogen ions being pushed across to create electrical potential. Here one goes, and then you can see the energy. So we're trading the energy in these high energy molecules, NADH and succinate and FADH, for energy from the electrical potential difference created by pumping H plus ions. And if that all sounds very complicated, it's okay. Take a deep breath. You don't have to know this. You don't have to memorize this. I just want you to appreciate the complexity of what's going on in almost every eukaryotic cell, in your body and every other eukaryotic organism. While we're here with the model running, let's look at a couple of questions that are on the lab. Which molecule is carrying electrons with the highest energy? So we're supposed to look for stars with the most points to figure out which electrons have the most energy or up on the top of those energy levels. It looks like to me that the NADH have the stars with the highest points, highest number of points. So let's look. We're correct. NADH is the molecule carrying electrons with the highest energy, which can be seen by the many pointed stars. So here's our high energy electron, and later down the electron transport chain, you're going to see some lower energy electrons, which the next question is about. Which molecule is carrying the electrons with the lowest energy? So we're looking for these very low energy electrons here that only have 3 points. You might be able to see them a little bit better here as well. So, what molecule is this here? It's got 1 oxygen and 2 hydrogens. 1 oxygen and 2 hydrogens we can abbreviate as H2O for the 2 hydrogens and 1 oxygen. And most of us know that as water. Let's see if we are correct. Yeah, those low energy electrons are hanging out on water. Let's go back to the lab sheet for a moment and kind of catch up. we saw which high energy molecules become part of the electron transport chain. And now we have to think about how does the electron transport chain use those high energy molecules generated in the Krebs cycle. Remember, we looked at the electrons kind of being traded around, those blue stars, and the electrons were losing some energy, but that energy was being transformed into an electrical potential. We were pumping H+ ions across the membrane, up the concentration gradient. So what we trade off in losing energy of the electrons from those high energy molecules here, we gain an electrical potential between the H+ ions pumped across the mitochondrial membrane. And if you need a little recap of that, it's right here as screenshots here. I also included what each of these protein complexes does. So you can read to your heart's content about all those protein complexes. And they'll become important here as we go to the next section. We're going to talk about what factors inhibit our mitochondria. We talked a little bit in class how poison can really mess you up because it messes with your mitochondria, and we'll see exactly how that works. So for folks who are following along in the application, I know some of you can't get the application to work, and that's okay. You'll get everything you need from the worksheet and from this video. But our big question here on page 7 is what factors can disrupt the production of ATP. Let's go down and take a look. We've got a couple of different options if we want to sort of mess with our model here. We can stop breathing. That'd be bad because we don't have any more oxygen here that shows up to pair with hydrogen. We can add cyanide into the mix, which is a poison, or we can add rat poison. So let's run the model and let's add rat poison and see what happens. So now here, this complex 1 can't receive high energy electrons from NADH. Bad news, because those are the highest energy electrons, and now we can't do anything with them because rotenone, this rat poison, is blocking that site. We still can make a little ATP. We can still pump some H+ ions across because succinate can land right here. We'll watch it happen in a second. Protein complex 2 is not inhibited, but we're not going to make very many ATP if we ingest a lot of rat poison, or rats ingest a lot of rat poisons. So don't eat rat poison. It's going to slow down your mitochondria because they're not going to be able to use their most high energy electrons. They're going to be stuck just working with complex 2 here. And you can see there's not a whole lot of action at complex 2 because there's not as much succinate going around. So here I'm running the electron transport chain as if we've stopped breathing and our body isn't bringing oxygen into our cells anymore. You can see that the electrons aren't traveling like they did before to bind with the oxygen here because there's no oxygen. And so the energy from the H+ concentration difference is not as much as it was. And so we're not going to be able to use those H+ ions to produce ATP in the next part of the process. We don't have oxygen here to take up those electrons when they get transported into protein complex 4. Cyanide also kind of messes with your cells, messes with your electron transport chain, and that it occupies the site where oxygen would normally come in. In this situation where an organism has ingested some cyanide, it's still breathing, it's got enough oxygen, but the site where the oxygen would come in here and pick up some hydrogen ions to make water is blocked. So before we answer any more questions on factors that can inhibit the electron transport chain, let's take a moment to go back to our lab sheet and look at the questions that we've answered. We know which molecule is carrying electrons with the highest energies. Those blue stars with the most points, look for where those are in the model. Which molecule is carrying electrons with the lowest energy? Those are the stars with the least number of points, and we saw them kind of in the lower right-hand corner of the model. What's happening to the concentration of hydrogen ions on both sides of the membrane? Well, let's go back and look here. The membrane here, the protons are being pushed out this way. Here's a better view. The protons are being pushed into the intermembrane space. You can see that's blue right here in this part of the model. And they're decreasing here in the matrix. So let's go back down and look at the question. They're being pumped out of the pink part and into the blue part, which is the intermembrane space. So they're going down in the pink and up. So we know the pink part, the matrix, the concentration is decreasing. We also know the concentration is increasing in the blue part, the intermembrane space. So it's decreasing in the matrix and increasing in the intermembrane space. And we're pumping those H+ ions to create electrical potential difference. We're trading the energy in the electrons from those high energy molecules we made in the Krebs cycle for an electrical potential difference across the membrane. Cells are pretty complicated, aren't they? So the decrease in electron energy results in an increase in electrical energy in the concentration difference. So now let's get back to poison. So you can see how cyanide blocks the place where oxygen attaches to protein complex 4. If you watch the blue electrons in the lab, how would oxygen not attaching itself affect the electron transport chain? Well, if we watch the electron transport chain, we saw that those low energy molecules get transferred to water. And if we don't have oxygen, we can't put the protons into water and transfer those electrons to kind of finish the electron transport chain. So that's pretty serious. If you stop breathing, so you don't have any oxygen, or you ingest a whole bunch of cyanide and you disrupted the electron transport chain of all of your mitochondria, life is going to be pretty sad for you. In fact, you're not going to be alive too much longer if you take a lot of cyanide. There's also a GIF file if you have a hard time getting the application to work. It's in the Dropbox for this assignment. You can look at how rotenone, rat poison, affects the electron transport chain as well. If the organism ingested rat poison, would it die or not? Well, it is poison. Eventually, the organism is probably going to die, but it might live for a while because it can still process succinate here in complex 2. So now to the finish line. This is the last part of cellular respiration. This is the part where we actually get to use the difference in the proton or the H+ concentration to make some ATP. And it happens in this really cool looking molecule called ATP synthase. The protons go through ATP synthase and help ADP hook up with a phosphate to get ATP. Let's take a brief look at this ATP synthase in action. Here's a YouTube video that illustrates how ATP synthase works with the H+, also known as proton gradient, to make ATP from ADP.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP.
You can see the ADPs and the phosphate coming in here and coming out as ATP.
The F1 ATPase portion of ATP synthase has been crystallized.
There they are. They're the reactants, and there's the product. There are reactants, and there's the product. So this is an enzyme catalyzing ADP, the reaction of ADP with a phosphate to make ATP. The final goal of this whole shebang, the whole cellular respiration. The animation we just saw is going to help you answer question 18, is what eventually happens to the energy, the potential energy, by pushing protons or H+ ions to one side of the membrane. We just watched that enzyme work, the rotor turn as protons kind of come back through the membrane, turn that rotor, and drive the conformation of the enzyme that catalyzes ADP hooking up with another phosphorus group to create ATP. So let's finish up here. Question 19, which of the following statements is true regarding the ATP molecule? Well, we know ATP is made from ADP and phosphate. We just saw that in the animation. ATP is a high energy molecule, but not because of its strong bonds. Remember, it's a weak bond between the second and third phosphate group. So B isn't true. Most ATP is directly made in the electron transport chain. Energy from a high proton concentration is used to make ATP. This is true. This is not, the ATP doesn't actually get made in the electron transport chain. That's where we create the electrical potential by pumping those H+, those protons, across the membrane that then flow back in ATP synthase to catalyze that reaction. Which of the following statements is true regarding the flow of energy in biological systems? Energy is constantly being lost. No, energy is never lost. It's just converted from one form to another. Sometimes energy is lost, sometimes it's gained. Protein concentrations always go from high to low. Nope, it goes both ways. If energy is lost in one form, it's just converted to another. It may be hard for us to sort of find it because it's dispersed in the system, but it's never gone. Energy is never gone. It's just transferred to another form of energy. Which statement is true of enzymes? Enzymes are necessary for each step of glycolysis and the Krebs cycle. Well, we saw that purple name of something between every step of glycolysis and the Krebs cycle. So we know this is true. This can't be true if A is true. This can't be true if A is true. And D can't be true if A is true. A is definitely true here. So our next question is, what role does the Krebs cycle play in the making of ATP? So we get pyruvate from glycolysis, and we do the Krebs cycle, and remember all those little yellow flashy molecules we made, those high energy molecules? That's the purpose of the Krebs cycle. we want to make those high energy molecules NADH, FADH2, and succinate and GDP. When we take those high energy molecules to the electron transport chain, we trade around some high energy electrons in those high energy molecules to move protons or H+ ions across the inner mitochondrial membrane. And those protons, that electrical potential, by pumping those ions, those H+ ions, are also known as protons, across the membrane. Remember, that's what we just watched in the video of ATP synthase. Those protons run the rotor of ATP synthase that changes the conformation of the enzyme at the bottom that hooks up ADP to ATP. So the electron transport chain creates the electrical potential that runs ATP synthase. And for question number 24, you get to use what you've learned in the previous 23 questions to connect all of these terms in 2 to 4 sentences. Please do highlight or italicize the terms as you're using them in your 2 to 4 sentences. Don't skip this question. It's worth 10 points. At least try to write something. Thanks so much. This is Miss R signing off on Lab 3.19, Cellular Respiration Part 2.